In this lecture, we'll learn what happens to the messenger RNA transcript synthesized during transcription. The central dogma, if you will recall, describes the flow of genetic information in cells. Genetic information is stored in the DNA, in the sequence of nucleotides found in genes. When the product of a gene is needed by a cell, a copy is made of the information in the form of RNA. This process, if you will recall, is transcription. If the copied information contains the recipe for making a protein, the RNA is read by ribosomes and a protein is produced. This process is translation. Let's review a few more details. Here is a general diagram of protein synthesis in a eukaryotic cell. How can you tell it's a eukaryotic cell? Right, it's got a nucleus. During transcription, the nucleotide sequence in a gene specifies the nucleotide sequence in a complementary RNA molecule. For protein encoding genes, the product is a messenger RNA molecule that exits from the nucleus and enters the cytoplasm. During translation, the sequence in a messenger RNA molecule specifies the amino acid sequence in the protein. Okay, now let's look at the details of translation. We'll concentrate on prokaryotes, but eukaryotic translation is very similar. The wonderful ribosome is the site of protein synthesis, as the messenger RNA binds to the ribosome and the protein is constructed at the ribosome. Just look at this cute little ribosome. Ribosomes themselves contain RNA and protein. The ribosomal RNA, or rRNA, is transcribed by RNA polymerase, but it is not translated. I wonder where that ribosomal RNA is made in the nucleus. Ribosomes consist of two subunits, the small subunit and the large subunit. In prokaryotes, the small subunit is called the 30S subunit, while the large is called the 50S subunit. Transfer RNA, or tRNA, is a single-stranded RNA molecule that brings specific amino acids to the ribosome for whatever the codon is in the messenger RNA. As you can see, the molecule folds in multiple locations because its nucleotides base pair with one another. Just like ribosomal RNA, transfer RNA is transcribed, but not translated, from genes in the DNA. Now, the transfer RNA contains an anti-codon, so it will bind to the correct codon in messenger RNA. An enzyme called aminoacyl tRNA synthase adds the correct amino acid to the transfer RNA based on its anti-codon. Here's a model of a charged tRNA. When the tRNA is bound to an amino acid, it is most correctly called an aminoacyl tRNA. That's kind of a mouthful, isn't it? Okay, let's just call it a tRNA. So when tRNAs are mentioned in this lecture, just assume it is a charged tRNA unless otherwise informed. And as this diagram shows, the transfer RNA is complementary bound to itself, forming a 3D structure. The 3' end is where the amino acid is bound. In this case, you see that phenylalanine is bound to this transfer RNA. That means that the transfer RNA must have an anticodon to recognize the codon for phenylalanine. The anticodon AAG corresponds to the codon UUC. If you look at your genetic code table, which I'm sure you have handy, UUC does code for phenylalanine. But wait, there's more. In addition, there will be another transfer RNA carrying phenylalanine. Which one do you think it will be? Yes, there is also a transfer RNA that recognizes the codon UUU in messenger RNA. Therefore, this transfer RNA must have an anticodon that will base pair with the other codon for phenylalanine. Our transfer RNA must have an anticodon of AAA to recognize the codon UUU. Let's now take a look at the process of translation. Initiation of translation begins with the binding of the messenger RNA in the small ribosomal subunit in the cytosol. Then a transfer RNA carrying the amino acid methionine binds to the start codon AUG. Notice that the amino acid has the anticodon of UAC so that it may recognize the codon. Then the large subunit of the ribosome binds to the messenger RNA, locking the transfer RNA into the P site of the ribosome. You can see that the next codon on this messenger RNA is UUC, so the next transfer RNA that binds is carrying phenylalanine. It enters the A site of the ribosome. The ribosome then catalyzes the formation of a peptide bond between the first and second amino acids, in this case methionine and phenylalanine. After the peptide bond is catalyzed, the messenger RNA is moved in a ratchet-like movement through the ribosome. This translocation is accomplished by proteins called elongation factors. The two transfer RNAs are moved in the ribosome when translocation occurs. Boing boing. The transfer RNA that was carrying methionine enters the E site and is booted out of the ribosome. At the same time, a new transfer RNA enters the A site and binds to the third codon. In this case, the codon is CGA. The transfer RNA that recognizes this codon is carrying the amino acid arginine. The ribosome catalyzes a peptide bond between phenylalanine and arginine, and translocation occurs again. Then another transfer RNA will bind to the codon in the A site, and this keeps happening until a stop codon, such as UAG, is reached. There is no transfer RNA for the stop codon. Instead, a release factor will bind to the stop codon. This results in the hydrolysis of the bond between the last transfer RNA and its amino acid, releasing the polypeptide and the transfer RNAs from the ribosome. Then the large and small subunits of the ribosome detach from the messenger RNA. The messenger RNA may be used for several more times before it is degraded or otherwise inactivated. Okay, let's put it all together in one diagram. The messenger RNA binds to the ribosome, and transfer RNAs complementary bind to the codons in the messenger RNA, reading it in a 5' to 3' direction. Peptide bombs are made between the amino acids carried by the incoming and outgoing transfer RNAs, forming the polypeptide. When the stop codon is reached in the messenger RNA, a release factor binds and the ribosome disassembles, ending translation. Now let's put it all together one last time and note some differences between eukaryotes and prokaryotes. Protein synthesis consists of transcription, in which an RNA copy is made of a gene in the DNA, and translation, in which ribosomes and transfer RNAs decipher the messenger RNA code and synthesize a protein. The RNA transcribed in prokaryotic cells has no introns, so it doesn't have to be processed. Also, since prokaryotes have no nuclei, there is no barrier separating the DNA and ribosomes. Therefore, transcription and translation can occur at the same time. But the RNA transcribed in eukaryotic cells does contain introns, so it does have to be processed. Also, eukaryotes do have nuclei separating the DNA and ribosomes. Therefore, the RNA must be modified to survive its trip outside the nucleus.